All right, let's dive in. Today, we're talking about a discovery that could literally rewrite first-year pathology textbooks. I mean it. This is a brand new, previously unknown way for a cell to die.
So for all you medical students and biology buffs out there, you probably feel like you have cell death pathways down cold, right? You crammed them for exams, you know all the players, but what if I told you there's a new member of the club? I mean, we all learned the big two. On one hand, you have apoptosis. That's the clean, programmed cell suicide. It's elegant, quiet, kind of like leaves falling in autumn. And on the other hand, you have necrosis, the total opposite, the messy, traumatic death. Think of a building collapsing because of an injury. Simple, right?
Well, you know, over the years, that picture's gotten a whole lot more complex. We've added players like pyroptosis and ferroptosis to the roster, but it turns out the cell's suicide squad is even bigger than we thought. There's a new recruit, and its method is, well, it's unlike anything we've ever seen before.
So let's meet the newest member of the team, Mitoxyperilysis. Yeah, try saying that one 3 times fast. Mitoxyperilysis. And this isn't just another variation on a theme, folks. This is a fundamentally new self-destruct button inside our cells.
And the name itself basically tells you the whole story, which is super helpful. Let's break it down. Mito for mitochondria, of course, the cell's power plant. Oxy for oxygen, but we're specifically talking about the toxic kind, reactive oxygen species, or ROS. Peril for the state of danger or stress the cell is in, and lysis, which means to break down or die. So you put it all together. It's mitochondrial death caused by oxygen radicals during the state of peril. Officially, mitoxyperilysis is a special kind of programmed cell death. But here's the absolute key. It requires a fatal combination of 2 very specific things happening at the exact same time: inflammation and metabolic starvation. It's not one or the other. You have to have both.
So this isn't some random accident. The cell has what's basically a 2-hit kill switch that has to be activated, kind of like a failsafe on a self-destruct sequence. So let's look at exactly what those 2 hits are.
Okay, hit number 1 is inflammation. This is the alarm going off. The cell's toll-like receptors. They detect a threat. Maybe it's a bacteria or a virus. It immediately kicks its immune response into high gear, getting ready for a fight. But as you know, that fight requires a massive amount of energy.
And that brings us to the second, simultaneous hit, starvation. At the exact moment the cell is trying to fight for its life, it gets cut off from its fuel supply. It's suddenly deprived of crucial amino acids like serine and arginine. So it's all revved up, ready to go, but there's no fuel in the tank. And that is when things get really, really desperate.
To really wrap your head around what happens next, we're going to use a simple analogy. Let's just imagine our cell is a bustling factory. This is honestly the best way to visualize the total meltdown that's about to happen.
So, here's our factory, a healthy cell. Everything's running like clockwork. The production lines are moving, the lights are on, materials are coming in, business is booming. Then, bam, hit number 1. The workers go on strike. This is our inflammation. The cell's internal machinery gets signals that there's a major threat. Everything grinds to a halt, chaos starts to build up, and the whole factory goes into crisis mode, trying to deal with this internal problem.
And at that exact same time, hit number 2 lands. The fuel trucks just stop arriving. This is the metabolic starvation. Now the factory is totally cut off from all the raw materials and energy it needs to function, let alone handle a crisis.
So, what happens? The factory manager, which in our cell is the lysosome, the recycling center, makes an absolutely desperate call. To keep the lights on, it decides to attack the factory's main power generator, the mitochondria. It literally starts tearing the generator apart just to burn its components for a tiny, tiny bit of emergency fuel.
But here's the thing. You can't just rip apart a power generator without some serious consequences. As the lysosome is shredding the mitochondria, the generator basically explodes. It unleashes this massive, toxic cloud of reactive oxygen species. That's our ROS leak. It is a catastrophic failure of the factory's core power system. And the result is inevitable. That toxic cloud of ROS is completely lethal. It corrodes everything it touches, dissolving the factory walls from the inside out. The factory is completely destroyed. The cell dies. And that right there is mitotoxopyrolysis.
But this isn't just some cool piece of cell biology. This discovery has absolutely staggering clinical implications. We've seen how it works, but now let's talk about why it matters so much. And the answer, really, is one word: cancer.
Now, this connects directly to oncology because a solid tumor is the perfect storm for Mitoxyperilysis. Think about it. The tumor microenvironment is naturally in a state of high inflammation because the immune system is constantly trying to attack it. And because tumors grow so ridiculously fast, they outpace their own blood supply, leaving the cells in the center starving for nutrients. They have both hits, just naturally.
So here's the crucial insight. Cancer cells have figured out ways to cheat this system and avoid this self-destruct pathway. But if we can develop drugs that block their escape routes and actually force them down the path of Mitoxyperilysis, we can essentially trick tumors into committing mass suicide. We wouldn't be poisoning them, we'd just be giving them a little nudge over the edge they're already standing on.
You know, this all points to a fascinating new way of thinking. For decades, we've tried to kill cancer from the outside, but maybe the answer is about reminding it how to die from the inside. This discovery opens up an entirely new front in the war on cancer, and it leaves us with a really powerful question. Could the future of cancer treatment just be about teaching tumors how to die?